time we have started, or we'll continue our lectures with regard to the action potential. We will see today how we are generating action potentials at neural cells. Okay, I think you know now the neural cells are also excitable cells, and they have the ability to generate action potential. Actually, what's the function of neural cells? Simply is to generate action potential and to get this action potential transmitted toward terminals. At the terminals, what happens? We are getting release of neurotransmitters, okay, from the terminal. So that's the simple function for the neurons. And we have that structure for the neural cell. We said we have cell body, we have long axon, sometimes that axon is up to half meter or more, okay? And we have some axons, as you see, are enveloped by what we are calling a myelin sheet. Actually, what the myelin sheet is a cell. That's the nucleus of this cell. That's nucleus here, nucleus, which is wrapping around the neuron to form this myelin sheet. And leaving, not all parts of the axons actually are enveloped by that sheet. We are having a small space here between these sheets, okay, where we have the membrane of axon is exposed to extracellular fluid. And these parts which are exposed to extracellular fluid, they are called nodes of Ranvier. You will know what's the importance of having these nodes of Ranvier. In addition, we have this part between the cell body and the axon, which is called axon hillock, which is having high functional importance in motor neurons, okay? At that site, we are getting generation of action potential, actually. In addition, we have a lot of dendrites. You will see the importance of these dendrites to increase the surface area of these neurons for many purposes, actually. In addition, we have a lot of terminals, as you see. From one axon, we can have many terminals, and in these terminals, we have neurotransmitters, which are stored in vesicle. Once we have action potential formed at this axon hillock here, we are getting this action potential is propagating along that axon to reach the terminal. So finally, this action potential will cause release of neurotransmitters. Now these cells which are enveloping axons, here in this type of neurons, these are Schwann cells, called Schwann cells. So they are part of what we are calling supportive cells. We have many types of supportive cells. I will not discuss these types actually in the class. Maybe you will be taking them with Dr.mohammad. We'll be talking about these Schwann cells and the neurons again, classification of neurons. Also, I will not talk about the classifications and so on. Okay. We have, a lot of functions for these supportive cells. I will not ask you about their names and so on. But the main function is to keep the media around the neuron very clean media. They are performing a lot of functions actually. One of the functions, they are having phagocytic ability. What do you mean by phagocytic function? So they can, anytime we have a pathogen invading neural tissue, these cells can engulf that pathogen, destroying that pathogen and so on. Secondly, we are needing to keep very low concentration of potassium in the media around the neuron to keep its functionality, the performance of this function to be the highest possible. How we are doing that? We have also these supportive cells can uptake that potassium, okay, to keep low concentration of potassium around the neurons. In addition, you will see that we have a lot of terminals around these cell bodies, for example, and these are releasing neurotransmitters. We are needing to keep the concentration, the lowest possible concentration of these neurotransmitters which are released by terminal as low as possible. How we can do that? We can have some of these cells uptake these neurotransmitters, destroying these neurotransmitters, recycling the components of these neurotransmitters and sending these end products to the neural cells to reuse these, for example, products, okay, and so on. So we can have a lot of functions for these supportive cells. One of the functions also, they are, you know, the neural cells are living for all our life, okay, they are not dividing. So we are getting release of what we are calling neurotrophic factors from these supportive cells to get or to increase the survival of these neurons as long as possible. We are getting these neurons living for all our life, 70 years, 80 years, 100 years, okay? How we are getting that? Because of the ability or of having release of neurotrophic factors from these supportive cells to support the viability of these neurons. Okay, so a lot of functions we have by these supportive cells. In addition, you see that we have dendrites. Okay? Actually, why we are needing these dendrites? A lot of them. To increase the surface area, to enable these neural cells capturing as much as possible of these neurotrophic factors to keep their survival. You will see also that we have terminal synapses. Most of the synapses are with the cell bodies, not with dendrites, actually, okay? So we are getting these synapses here with the cell body. So the dendrites are not having the function as for the synapsing or for communication between the neurons. Mainly, they are for capturing needed products from the media around neurons. Okay. So, assuming we said the function for neural cells is to generate action potential. Assuming we already have generated action potential, we will see how actually we are generating that action potential. And in motor neurons, we said we are generating action potential at the axon hyper here, at the junction between the neural cell, okay, the cell body actually, and the axon. Now, once you have an action potential already generated, what is the potential of that membrane? It will be positive where? Inside and negative outside, okay? Now, once you have situation positive inside and negative outside, here the nearby region is still polarized, is still at resting potential. So what we mean by that, it is negative inside and positive outside. Now you have two nearby regions where you have positive charges and negative near it, and inside negative and positive. Okay? Now what happens? We are developing currents actually between the positive with the negative. You know how we can get the depolarization process by these currents. Okay? Currents inside and currents outside the membrane. By these currents, we are getting depolarization of that region. Once you have depolarized that region, reaching the threshold, this region now is developing that action potential. The same thing happens with the nearby region here. Currents causing depolarization. Once you have reached the threshold, developing a new action potential in that region. And so on. So in these unmyelinated fibers, this is myelinated fiber, and these are unmyelinated fiber. if you are viewing the action potential spreading along axon, is spreading in a continuous way. And because of that, if you are viewing it in series, okay, we are calling this type of conduction along these unmyelinated axons as continuous conduction. Again, as you see, we have currents developed. More important are the internal currents than the external currents, internal, okay? By these currents, we are getting depolarization of that region. So this is the region where you have an action potential, for example. You are developing currents, and by these currents, you are depolarizing this part of the membrane, reaching the threshold, generating action potential, then going to the next part of the axon and so on. So we are getting like a continuous conduction along that the membrane of the axon. As you see here, we have also currents between the region where you have action potential and the previous region. When we are not developing an action potential here again, the previous region.
Refractory period.
Because we have refractory period. We have that region and refractory period. So what's the status of sodium channels in refractory period? They are
Inactive. Not
closed
capable.
and not capable for opening. Okay. So these currents, they are not able to cause these channels to reopen again. And this is one actually of the functions of this refractory period, to get propagation of action potential in one direction only. Okay, we are not having that action potential going back and forth over the membranes of the axons. So once it's developed in this part, it's traveling toward the terminal, okay, of that axon. And that's the importance of the refractory period, that we are having to get propagation of action potential in one direction only. So this type of conduction again in unmyelinated fiber is called continuous conduction. Again, these are the currents. We said most important are internal currents in causing that depolarization. Once you have that region is in action potential, by these currents you are developing, depolarizing the membrane, reaching the threshold, and developing action potential at this part of the membrane, then at the other part, and in series, you are finding that action potential is propagating along that axon at specific velocity. Now in myelinated, what happens? The same thing actually. We have at the axon hillock here, an action potential developed. We have the myelin sheet, but we have a part of the membrane here which is exposed to the extracellular fluid, polarized as you see, so it is negative inside, positive outside. But the axon hillock is positive inside with regard to outside. So the same thing, we are developing currents actually between the axon hillock internal currents here and the first node of Ranvier, and by these currents we are depolarizing that membrane. By depolarization, reaching the threshold, generating action potential and so on. Then going to the next node of Ranvier, currents, okay, between the nodes of Ranvier results in depolarization and so on. But if you are viewing the action potential which is generated at the axon hillock here, it's like jumping from here to the first node of Ranvier, then to the second node of Ranvier, and third node of Ranvier, and so on. Jumping or salting. And because of that, we are calling this type of conduction as saltatory conduction. Saltatory, okay? So saltatory conduction is taking place in myelinated, while the continuous conduction is in unmyelinated. Secondly, which of these is faster?
Myelinated.
And myelinated, the saltatory is faster because we are skipping big parts of the membrane. Here, are you getting action potentials in the region which are covered with myelin sheath?
No.
No. Only at the nodes of Ramsey, as you are seeing. Now, if you have, this is what happens actually, action potential here, causing currents, the next node of Ramsey results in depolarization, generation of action potential, then currents, and so on, okay? We are not getting a back here, okay? These back currents, they cannot cause the sodium channels to reopen again. So this is in the refractory period. Now, we said we have faster conduction in myelinated, but if you have two unmyelinated fibers, nerve fibers, once you are hearing about nerve fibers, we are talking about action, okay, of neuron. Now, if you have two nerve fibers, both of them unmyelinated, one with bigger diameter, one with a smaller diameter, where you have faster conduction, bigger diameter or smaller diameter fiber?
Bigger.
Huh? Bigger. Why? Hmm? Because we have lower resistance for the internal currents. You remember, if you have smaller fibers, you have higher resistance for ions to move. If you have bigger diameter, you have lower resistance for this ionic currents inside. And because of that, I told you more important is the internal current than the external current, okay, to get the conduction. So it will be faster in larger fibers and slower in smaller fibers. And because of that, you will see with Dr. Fatma differences between the categories of neurons. We have alpha type, for example, C fibers and so on. Depends on the types of these neurons, if they are myelinated or unmyelinated, larger diameter, smaller diameter, fibers and so on. Okay, again, we said at motor neurons, we are generating action potentials at the axon hillock. What about sensory neurons? What they are doing, sensory neurons? Sensing information from the body, sending this information toward the central nervous system. Are we having cell bodies in our body? Sorry, in our periphery, for example?
No.
Muscle? Skin? No. What have? Actually, we have terminals of a sensory neuron. The cell body is located in the spinal cord. Terminals, but these terminals are equipped with receptors, which are sensing something. Receptors sensing changes in temperature. We have receptor temperature sensors, for example. Touch sensors. If you are touching something, you know what you are touching, okay? And so on. So these sensors or receptors are generating action potentials. Now the action potentials are transmitted toward the central nervous system, not toward the cell body. We don't need that. Cell body does not need this information. The action of these types of cells for the first neurons actually is like T letter, T. So you have from the receptors here, action potential is traveling along the axons, and to have then the second part of the axon, we have terminal, and these terminals are synapsing with the second cell, sending information, then your nervous system is processing this information, and so on by your neural network structure, okay? So in that way, we have differences. So finally, we have here terminals where we have neurotransmitters, and these terminals are releasing that neurotransmitter. How? I think you remember once we have talked about vesicular transport, we said we have vesicles here filled with neurotransmitters. Once we have action potential reaching these terminals, causing more vesicles to look with the membrane. How that happens? Simply by activation of calcium channels. What type of calcium channels we have here? Voltage gating channels. We have, since the change in voltages, activating these channels, we are getting entry of calcium. The entry of calcium causing more docking of these vesicles with that membrane, okay? And by docking, we are getting the release. The whole structure here, it is called synapse. Synapse. The membrane which is belonging to the terminal here, it is called presynaptic membrane. Presynaptic, okay? Now, once we have the release of neurotransmitter from the terminal, is released into a small space here, which is separating two membranes. Actually, the second membrane here is belonging to the second neuron, and we are calling it as postsynaptic membrane. Okay. We have different structure, as you see what we have over the postsynaptic membrane.
Receptor.
Receptors for that type of neurotransmitter. Now the receptors. You are finding some receptors are linked to sodium channels. If we have a receptor linked to sodium channels, what you are getting as response by the second neuron?
Action potential.
Activating sodium channels, what we are getting? Depolarization. If you have the receptors linked to potassium channel, what you are getting?
Hyperpolarization.
Hyperpolarization. Okay. Now these are developed at the postsynaptic membrane, and we are calling these potentials. They are just depolarization, not reaching the threshold. We are not reaching the threshold. They are not action potential. Okay? So we have only small depolarization or hyperpolarization. These are called postsynaptic membrane potentials that are classified as either excitatory postsynaptic potentials or inhibitory postsynaptic potential. What do you mean by excitatory postsynaptic potential? The potentials that are developing what?
Depolarization.
Depolarization. Okay? From now on, we are calling them as excitatory postsynaptic membrane potential. They are developing at the postsynaptic membrane. What is the characteristic of inhibitory postsynaptic potential? Simply, it is hyperpolarization, and they are called in that way. So this is the mechanism. What happens at the terminals to cause the release of neurotransmitters, and these neurotransmitters now binding to a specific receptors, activating specific channels. By activation of these channels, we are changing the membrane potential or the potential of the postsynaptic membrane to either, you know the mechanism, okay? This is, for example, acetylcholine receptor is linked to sodium channels, which can cause depolarization of that membrane. And as you see, we have a lot of terminals that are synapsing with the cell body of the postsynaptic neuron. You know now what we mean by postsynaptic neuron. So the second neuron, okay, called postsynaptic neuron. Are all these terminals causing excitatory potentials?
No.
Are all these terminals causing inhibitory potential? You may find terminals from that neuron, for example, causing excitatory potential, but other axon with many terminals, they are causing inhibitory potential. So now the job of the neuron to decide to generate an action potential or to generate no action potential. How that happens. Okay? As you see, we have potentials only, like this one, which are excitatory postsynaptic membrane potential, or hyperpolarization like this one, which are by terminals, which are causing inhibitory postsynaptic potential. Now we have a process of what we are calling summation, summing. these events together, okay? If the sum of all these events at the axon hillock, they are summing and migrating along that membrane, okay? This will unify with that, and so on. But the migration is with different rules than the rules for the migration of action potential. The action potential, is keeping the same strength, okay, along the axis, because you have activated threshold. Here we are not having threshold. You are starting, for example, with higher amplitude of this excitatory potential. After a distance, you have lower amplitude, and so on, okay? Now, once these are meeting with each other, they are summing with each other to get a higher, lower, higher, excitatory meeting with inhibitory, maybe you are getting lower, okay? Excitatory and excitatory are getting higher. Now the decision is taking place with at the axon hillock. If the sum of all these potentials, by that sum, you have reached the threshold, okay? At that part, we have plenty of voltage-gated solutions, more than at the rest of the cell body. So if the sum, by that sum, you have reached the threshold, you are generating action potential. If the sum of all these events, or by that sum, you are not reaching the threshold, you are getting no action potential. So the decision will take place at this part of neuron, which is the axon hillock, to generate action potential or to generate no action potential. So this is representing like inhibitory post-synaptic potential. This is down representing like excitatory post-synaptic potentials. Now, to understand the process of summation, we said we can have some terminals causing excitatory potential like this one, another terminal here causing excitatory potential like this one, okay? If the two are met and they are at the same time, we are getting higher, okay, potential at the threshold, at the axon hillock to reach the threshold. Once you have reached the threshold, you are developing the whole action potential. Now what happens if excitatory meeting with inhibitory? Canceling each other, for example. Okay, we are not developing, in this case, an action potential. For the purpose of understanding, we have two types of summation. This type is called the spatial summation in space. As you see here, we have two presynaptic neurons. We have the neuron A and the neuron B. Okay, if you have stimulus only from neuron A, you are getting the small potential, small excitatory. From neuron B alone, also you are getting small potential. But if you have both of them, or the signal reaches the postsynaptic neuron at the same time, you are getting bigger, okay, potential, reaching the threshold, generating the whole action potential. Now, the second is called temporal summation, which is referring to time, actually. You have only one presynaptic source, as you see. So, synapsing with the second neuron, stimulus, one stimulus from that presynaptic source is causing small depolarization. Second stimulus is causing only one small depolarization. Now imagine that you have many stimuli coming one after another from the presynaptic neuron. So you are in depolarization, a second stimulus coming, causing more depolarization, third stimulus coming, causing more depolarization, and reaching the threshold. Once you have reached the threshold, you are developing an action potential. Pay attention to the time. Here for the action potential. It is much shorter actually than the time for the excitatory potentials. Okay? The time of excitatory potential is longer than the time of the action potential. And because of that, you can have frequency of stimuli coming from the first neuron to stimulate the second neuron. Actually, that's the job of our nervous system in processing information, sending commands according to the information that are coming. If we have many excitatory potentials coming to these neurons, we can get, or from many other sources, we can get reach the threshold and sending the command to final effector structures. Okay? Or in our nervous system, actually, we have more inhibition than stimulation, trying to keep neural cells quiet. Okay? So by more inhibition, once you are needing to stimulate that type of neuron, we are getting more signals, which are stimulatory signals, causing excitatory postsynaptic potential over these neural cells, and to generate these neural cells, now they can generate action potentials. So in that way, we have neurons are working. Sorry. The same thing here, as you see, we have when stimulus from one neuron, for example, alone is causing small depolarization, but if we have both of them here down, at the same time, we are reaching the threshold, developing action potential. Here, one excitatory with one inhibitory, they can cancel each other. We are developing more action potential or no change in the potential. Here, if you have, for example, the same, okay, many stimuli coming from the first source, from the same source, okay? So what you are getting, depolarization, more depolarization, reaching threshold, and generating the action potential. Over our neural cells, you can have both types of summation, the spatial and temporal summation together, according finally to these stimuli that are coming. So that second neuron now is deciding to generate an action potential or to generate no action potential. As you see, we have in our neural network structure a complexity of organization. You can find a neural cell like this one, axon, with many terminals. Each terminal is ending with another type of neuron. Not all terminals are ending with over the same neuron, okay? So this type of organization is called divergence in our neural network structure. Also, you can find cell body like this one in red, receiving inputs from many sources. And this is called as convergence in our neural network structure. You have also other forms, I think Dr. Mohammad will tell you about them, but more important now for us is the convergence and divergence to understand the complexity and the responses of the autonomic nervous system when we are talking about autonomic nervous system next week. Okay? Now, so far, we have recorded the potential, the membrane potential, and the changes in that membrane potential during action potential by placing one electrode inside and one electrode outside. So we have measured the potential differences over time. Okay, in the x-axis you have time, in the y-axis you have voltage changes. Okay? The question is, can we record the changes in the membrane potential by placing the two electrodes outside? Yes. Can we record? What will be the potential that can be recorded at the resting potential, okay, at the rest, if you have placed the two electrodes outside? Zero. Okay. Now imagine that under one electrode, you already have developed an action potential. So what you are getting? Positive inside or negative inside? Positive inside. Outside is negative. Now the second part, where you have the second electrode, is still polarized, so it is positive. Can we record a potential difference in that situation?
Yes.
Hmm?
Yes.
Yes, we can. Okay. So you can record a potential difference, which will be positive to say. Now, once you have repolarization, that part becoming near zero, or okay, more negative, more positive outside to say. Now the second part, where you have the second electrode, has developed the action potential becoming positive. Okay. Also, you can record a potential, but in that way you are recording a wave in the opposite direction. Okay. So the first wave which you are recording, it is called depolarization for the biphasic action potential. And the second wave is called repolarization. Okay, repolarization. Because in the first region, you have now repolarization. Okay, so you can record that by placing the two electrodes outside. You can record first a positive wave to say it is depolarization, then a negative wave to say repolarization, but you are finding in some literature that the first wave which can be recorded is a negative wave, and the second wave is a positive. Simply by switching electrodes, okay, the positive and the negative, if they have switched, you can record in the reverse direction. You have the first wave in the negative direction, but still we are calling that wave as depolarization. And the second wave in the positive direction, but still we are calling it repolarization. So we can record in that way the changes, or to say you have placed one of these electrodes or connected that one electrode with the high resistance. That means the voltage is zero. You can also record according to the zero voltage. Okay, what you are getting more than zero, which is in the positive direction, or in the negative direction, you can record that potential. These types of potentials, which can be recorded by placing the two electrodes outside, we are talking about recording of biphasic action potential. So far, what we have discussed with regard to the action potential that we have discussed, all that is referring to recording of monophasic action potential. So by having electrode inside and one electrode outside. Okay, so this is the difference between the monophasic and the biphasic action potential. Biphasic, simply we are placing the two electrodes outside the membrane. Now, is that important to get recording? Actually, we have some techniques by which we can record the whole action potential over the whole cable of nerve. Okay, you know the nerve is composed of many nerve fibers, thousands, okay, of nerve fibers. That's the nerve. Now, to say you have stimulated all that cable. Once you have stimulated all that cable, you can record the changes of the potentials, okay, which is giving you a summary of the sum of all action potentials now that can take place in that nerve or that cable. To say you are recording at that point, okay? So what you are getting at that point, that recording in this way. If you are recording at farther distance, what you are getting? The same waves that you have seen here have been split. Why we have them split? Same waves.
لأنه الإمبلس مش نفس السرعة.
The third recording, as you see, you are getting more split, for example. Farther distance, you are getting much higher splits, okay? Why you can have that?
لأنه في fibers myelinated في non myelinated وفي
You will see that we having the same velocity of conduction along all nerve fibers, okay? You have different fibers with different velocity of conduction. You have fastest, for example, fiber. These are fastest, which are characterized as alpha fiber, okay? They have fastest conduction. Now, as you see, you can record the activity that happens at the lowest, at the fibers with the lowest velocity of conduction, okay? Because you have needed more time, actually, in x-axis, you have time. You have needed more time to record the changes that happens in that group of fibers which are conducting at much slower rate. And you are getting what we are calling compound action potential. In this case, which could be useful for estimation the integrity of the function for a group of nerve fibers, okay? It's useful in clinical to summarize or to know how that group of nerve fibers are functioning well, which are, for example, innervating the hand, okay? Placing stimulators somewhere to stimulate all nerve fibers and placing electrodes at different distances to record these waves for the activity of all nerve fibers or all nerve fibers that are composing that nerve, okay? This is another recording, actually, as you see. So these are the fast fibers here, okay? At a further distance, you may find more types of waves according to the velocity of conduction. And this type of recording, again, they are called compound or recording of compound action potential. And according to that, you will see classification, actually, for the nerve fibers. And you will take these classifications with Dr.mohammad will tell you about what types of nerve that we are having, what types of neural cells. We have C fibers, alpha fibers, beta, gamma, and so on, okay? So according to that, we have classification and groups of these, and where we are finding also all this information, we'll take them with Dr. Mohammad. If you have questions. So next time, next Tuesday, we will meet at Yazar. At 12 o'clock, okay? يوم الثلاثاء القادم. انتوا والمجموعة أظن راح تكون مجموعة 5